most of the time we focus on neurons and synapses because, you know, the disease is mainly memory, affect the memory. So, but recently a lot of attention, and rightfully so, have been focusing on the immune system and astrocyte, glia, and definitely the neurons are not in vacuum. They are interacting with these other cell types. So one of my students wanted to look at these tau oligomers in astrocytes. And she did in human tissues, mouse tissues, and she found that they're there. But the key find, okay, for us, this is descriptive, but still important. Maybe it's like oligomers like they can be transported from neuron to astrocyte, vice versa. So that was the focus. But then the student made an observation that there is association between the presence of these oligomers and one protein which called high mobility group box 1, HMGB1 protein. And that was, and we proposed like a hypothetical thing, like HMGB1 and oligomers are associated. So this is how it started, the whole thing. And after that, a couple of groups published a lot about astrocyte senescence in tauopathy mouse models. And that was really exciting, but it took us a while to realize that HMGB1 actually is also part of the senescence-associated secretory pathway, the SASP. And then it kind of eureka moment, like, oh, maybe this is connected. And this is why we went on our journey to investigate the hypothesis, or at least to test if the oligomers or pathological tau trigger HMGB1 release. HMGB1 is normally in the nucleus, but if it's released into cytoplasm, then it becomes a bad signal. And this is what we looked at, and we show it in different models, in vitro, in vivo. So basically, that was a more mechanistic link between pathological tau and astrocyte senescence. And the problem with senescence is that once it starts in one cell, they secrete molecules that they spread in the tissue, like in the neighborhood. So that was pretty intriguing. And the other thing which we wanted to focus on, that senescence also a risk factor in aging. So basically, it was clear the more we age the animals, the more senescence we notice, or tau transgenic animals. So